organizers for the invitation. It's my pleasure to present our recent line of research about developing this simulator for single cell and spatial omics data. So our purpose includes benchmarking and model inference from data, and also to generate in silico controlled experiments. So first of all, let me briefly introduce the structure, a common structure of single cell spatial omics data from a statistical modeling perspective. The process data will be a cell by feature matrix. And by cell, I mean broadly, it could be a spot in spatial omics, which may be considered multiple cells jointly. But here I will just refer to it by a cell. And also a covariate matrix where the rows are cells, but we have multiple covariates to describe every cell. I will give some examples. So the cells may have some heterogeneity structure that are observed or not. So the structure could be discrete cell types or a continuous trajectory of cells, or their spatial locations. So this is known for spatial omics data. And for the experimental design that were used to generate the data, they may include batches, which are the effects we don't want, or conditions, which are the biological signals we want. And finally, we have multiple features nowadays. The most common one is gene expression. We have this for single-cell RNA-seq data or spatial transcriptomics data. Or we can have the chromatin accessibility in single-cell ATAC-seq data. And SNARE-seq is a technology that can give us both gene expression and chromatin accessibility. And we may also even have protein abundance data. So a rich data resource. And the motivation for our development is first, benchmarking computational tools. So as we know, this is a very popular field, and this website has a collection of the computational tools for single-cell RNA-seq data. There are already more than 1,000 tools up to date. And so from a user's perspective, how can we choose the most appropriate tool for our analysis? That's the question. And the second question is, can we do inference on these data? So here are some typical questions I'm listing. So conditional cell covariate. For example, the cell's type or the cell's pseudotime in the trajectory or the cell's spatial location. Can we infer every gene's distribution? And can we infer gene pairs correlation? And the gene can be replaced by other features. And finally, how can we design in silico controlled experiments? So this is motivated by real biology experiments. We have to use negative control, and sometimes we need to add positive control so we can evaluate the results. However, this is often lacking in computational pipelines. So we hope to offer a way to generate the controls to evaluate the results. So for all the purposes, we need a realistic simulator with interpretable parameters. So we started this journey in 2019 with this work ScDesign. So that one was designed for use a simulator to help optimize the trade-off between the number of cells and the sequencing depth from the perspective of detecting differentially expressed genes between two cell types. So the pros of this method includes that it's a probabilistic model with interpretable parameters, and we model cells as observations. So therefore, the cell number is the sample size. We can vary the sample size, and we can vary the total sequencing depth as the budget. So we can find a trade-off. And my former student used this method for benchmarking the doublet detection methods, the computational approaches, which doublet detection is a necessary step in single-cell RNA-seq data processing, and we have some interesting results. However, we also realized this ScDesign has cons. The biggest con is that it models genes as independent variables. Then that's okay from the perspective of differential expression because we look at genes one by one. But for other analysis, this is not okay because the cells are not that realistic. And also in ScDesign, we will first do a log transformation of the counts and then use normal distribution. And that's considered not as straightforward as modeling counts directly. Okay, so motivated by those limitations, we take a look at the existing simulators in the field. So I try to summarize them from different aspects. And I want to highlight this gene correlation capture. We can see that most methods couldn't do the job. And also most methods couldn't vary the cell number, sample size, or the sequencing depth. And many of them don't even preserve genes. So motivated by this, we have our version 2, ScDesign3, which uses a different formulation. So we want to model counts directly. And also we want to preserve gene-gene correlations by modeling the genes' joint distributions so that the cells will be more realistic. And we also want to preserve the advantages of ScDesign for allowing variable number of cells and varying sequencing depth for experimental design. And there are two related work also considers gene-gene correlation, but they have some differences. So this one from Catherine Roeder's group, they would allow users to specify marginal distributions of genes as input, while we will infer that from data. And this other method doesn't allow varying sequencing depth. Okay, so I will first describe ScDesign3 technically, briefly. So we would denote the gene matrix, count matrix, as a P by N matrix. Here, the rows are features, P genes. The columns are the observations and cells. We would assume that the cells are already divided into K cell types. And so we know which cell belongs to which cell type. So this is considered as prior information in the simulator. And then we will focus on the small K cell type. And we look at the submatrix with the corresponding cells as the columns and the P genes as the rows. So our goal is to fit a parametric probabilistic model for all genes expression in this cell type K. So for the simplicity of notation, I will drop the K notation in the following discussion and just focus on one cell type. So we want to model counts directly. Our first step is to model the marginal distribution of each gene I. So given gene I and I go from 1 to P, we have this count vector for cell J. So we consider each cell as a random vector. So this is one cell, p-dimensional vector. And we would use the small x to denote the observed count of gene I in cell J. So our goal is to select a marginal count distribution for gene I's count. And we would allow a different count distribution for different genes. So we consider 4 common count distributions: Poisson, negative binomial, and their corresponding zero inflated versions to account for the sparsity in single cell data. And for the joint distribution, which is the key part for our modeling, the P by P, right? How can we get the p-dimensional joint distribution? So we will use the copula framework in statistics to do the job. So the idea is that given we have each gene's marginal distribution, for gene I call this Fi. So for the P genes, we have F1 to Fp. And to link them or to couple them into a p-dimensional joint distribution, we need this copula function C. Okay, so with this, then we can have the p-dimensional joint distribution of the P genes. But the key result for the copula thing is that the P distributions, F1 to Fp, they must be continuous. So there's a unique copula function to define the p-dimensional joint distribution. However, in our case, the P marginal distributions are discrete. So we try to address this issue by using distributional transform, which essentially converts each discrete distribution into a continuous distribution by uniformly distribute the point mass between 2 consecutive discrete values into their interval. So to obtain this continuous distribution F1 to Fp, we apply this transformation. And then we use the Gaussian copula to couple the P marginal distributions into a joint distribution. And so in this Gaussian copula, the only parameter we need to estimate is this P by P correlation matrix. So with this, and then with the F1 to Fp, we estimated for the marginal distributions, we obtain a joint distribution for the P genes. Okay, so then the estimation procedure, we've done this in 2 steps. The first is to estimate marginal distribution for every gene. And then we apply this transformation technique to make the distribution continuous. And then we use the transform values to obtain the Gaussian correlation matrix. So essentially, we would apply the standard normal inverse transformation, CDF transformation, to every value here. And then we use the transform vectors to estimate this correlation matrix as the sample correlation matrix. So I will skip the generation part. So essentially, with a fitted joint model, and we can specify the cell type proportions, which can be estimated from real data or specified by users. And also, users can specify the number of synthetic cells they want and the total sequencing depth, which we will use to adjust the model parameters proportionally. Then we can obtain a synthetic gene by cell count matrix with the K cell types. And also, we can output the fitted model parameters for interpretation. So this is what we can see, right? This is data used for model fitting. This is the data reserved for validation. This is the synthetic data. And we can see copula plays a critical role. Without using that, the synthetic data would look like this and not look like real data. And these are two competing methods which use different formulation. And we put them here for comparison because they have some implicit modeling to account for gene correlations, but not as explicit as our framework. So we can see that their synthetic data are not very similar, not as similar as what we have here. And we also, this is a summary. So basically, we have a multi-gene probabilistic model for each cell type. And the key is to capture gene-gene correlations using the copula framework. So this is the work we published last year, and that's not yet. So we can see that here the limitation is that we are only generating synthetic data for discrete cell types. However, the cells may come from a continuous trajectory, right? So then we want to generalize this marginal distribution of gene I in cell J to be dependent on flexible cell J's covariates. And they may include the cell type, which is what we have already, or the cell pseudotime in the trajectory, or spatial coordinates, or the cell's conditions, or batches. We hope to incorporate all of those into our synthetic data generation. So to achieve that, we generalize that marginal distribution modeling to use the generalized additive model. So in this model, we can allow the negative binomial distribution for gene I, so that mean parameter can be a nonlinear function of cell covariates. So then we can incorporate covariates into the generation. So this is our mod version 3. So we call this ScDesign3, which is I call this Omnibus single cell spatial omics simulator. What it can achieve is that the cells can follow continuous trajectory in addition to discrete cell types. And the feature modalities has been generalized from RNA-seq to more features like open chromatin, protein, spatial coordinates. And also we can allow model selection by likelihood. So our simulator, after fitted to real data, can output a likelihood. Then we can use criterion like BIC for model selection. So this is a quick snapshot about what a new simulator can do. So this is the real data. This is ScDesign2, which will divide cells into clusters first. And ScDesign3 can fit a continuous trajectory. So more realistic than ScDesign2 for this data. And furthermore, we can see that this is the real data. We can generate data with a dividing, bifurcating trajectory. Top is one direction and the bottom is another direction. While in other simulators, they cannot really use the pseudotime as ground truth in generation. So in other words, they do not provide a trajectory for benchmarking. And of course, we can also generate data for benchmarking data modality integration. So like here, the cells here have RNA-seq only. The cells here have methylation only. So if we want to benchmark a method that does this cross-feature integration, we can generate cells that have both modalities and then use the integration method to do the integration and do the comparison. And this is what we can do for spatial data. And finally, my other student has developed a new functionality so that we can not just generate counts, but also the reads. So basically, here is the dimension reduction of cells using counts. This is real cells, synthetic cells, and this is the corresponding browser track for the reads. So being able to generate reads will allow us to benchmark the tools that does the low-level processing, not just counts. So here's just a figure showing what this new simulator can do. For the simulation part, it can generate data from different cell states and also different features and also different experimental designs. And for the application part, when apply the simulator to real data, we can infer the model parameters so to interpret our data. And we can also do model selection based on the likelihood. And also we can do model alteration. We can alter the model in a way that matches our null hypothesis, so we can generate the synthetic null data. So lastly, I want to say, why do we need in silico controlled experiments? So this is from the Khan Academy website. So we know that in the real biological experiment, we need a no negative control so we can really evaluate our experimental outcome. However, in single cell inference problems, it's very tricky because we have a pipeline that will analyze the same data for more than once. This is called double dipping. And so for example, one example is that we would often cluster cells first and then identify differentially expressed genes, DEGs, between cell types. So why is this problematic? We could imagine that if there's no clusters at all, we force the cells to be divided by expression data, then we could still find some differentially expressed genes between the forced cell clusters, and that may not be the thing we want. So there are 3 existing papers that discuss this topic. The first 2 use the so-called selective inference framework to obtain some closed form solutions for the statistical test, but they have to assume that the genes follow Gaussian distributions. And maybe just a week ago, also from Daniela Wooten's group, she posted another bioarchive paper, new archive paper, which doesn't require Gaussian distribution assumption anymore, but they will require to split every gene I's count in cell J, randomly splitting that count Xi into 2 counts. So I have to say, we didn't do a specific comparison with their approach yet, but from my perspective, they use a different formulation. So in their formulation, the cells are not observations, but fixed. So in other words, in their formulation, the cell number is fixed as n, while we cannot change the cell number. And that's what's different from our formulation. So our proposal is to use our simulator to generate a corresponding synthetic null, and then use another approach I'll describe very soon to achieve the FDR control. So the whole idea is inspired by the gap statistic for selecting number of clusters and the knockoff procedure for controlling the FDR, false discovery rate. So let's take a look at this data. This is the real data. So if we want to know whether there are 2 clusters or not, before we do the differential gene expression analysis, just doing a permutation of the cell type labels is not sufficient, because you can see that here in the middle, permutation will disrupt the labels, but the cell's shapes will be preserved. While using our ScDesign3 simulator, we could actually generate this null data that look like real data, but there are no more cell clusters. So this will serve as a negative control for testing whether there are 2 clusters or not. So the clipper is motivated by the knockoff procedure. And so the goal is to achieve FDR control without requiring high resolution p-values, because this is usually serve as the intermediate for FDR control, right? And also to achieve high resolution p-values, we would often require either parametric assumptions or large sample sizes to achieve the p-values. And if we require large sample sizes to do permutation-like tests, then the p-value calculation can be computationally intensive. So we want to avoid those troubles by using this p-value-free FDR control. So the goal is to do multiple testing with D features. And the difference between what we do here in the knockoff paper is that we are doing marginal feature screening instead of putting them into a high-dimensional prediction model. So in marginal feature screening, we examine 1 feature at a time. So the idea is to construct a contrast score, 1 per feature, and pulling the contrast score together. The theoretical condition for FDR control is that for the features that are from the null, their absolute value of the contrast scores and the contrast score signs would be independent, which should mean that the majority of the contrast score should be symmetry about 0. And only a small proportion of features should have contrast scores in this right tail. So this formula is trying to find the threshold here, the dashed line here, so that this ratio, which means the denominator is the number of features on the right of the dashed line, the numerator is the number of features to the left of this negativity of the dashed line plus 1. So you can consider this as an empirical estimate of FDR, but the theory can guarantee that by setting this T to satisfy that this ratio is less or equal than Q. So we try to push this dashed line to the left as much as possible. That is where we can set a cutoff, and then we can control the FDR. So you can see that we save the trouble of calculating p-values, but just need to construct the contrast scores. So this is our Clipper paper published last year, and applying that to this context, the cluster DE problem. So we can construct contrast score for feature J or gene J using this simple formula. So we just need to have our real data as the target and the null data generated as the negative control from ScDesign3, and apply a pipeline, the same pipeline T, to both data and then do the contrast. And the idea is that if this feature J is not interesting, then this difference will be equally likely positive or negative, regardless of its absolute value. So this is a very preliminary result of this idea. So suppose that this is the real data, which don't look like clusters, right? There's no clusters. And if we use ScRead clustering, the popular pipeline, or the standard classical k-means clustering, we can still divide the cells into 2 clusters. And visually, we can see that between the 2 clusters, we will be able to find some differential expressed genes. This is exactly the double dipping problem because the same expression data were used for clustering. So no wonder the 2 clusters will be different in terms of gene expression. But if we use our ScDesign3 to generate null data, we can see that it serves as a very good control, and it is similar to real data. So we can imagine that no DE genes should be caught from this comparison. And this is what we get. So for this example, if we run ScRead pipeline with its built-in statistical tests, many tests, they all have FDR equal to 1 because they will always find differential expressed genes between the 2 clusters. But using our approach, ScDesign3 plus Clipper, we can control the FDR very well. So the take-home messages include that we propose a simulator ScDesign3 for method benchmarking, parameter inference, which I didn't talk about, and also in silico control data generation. And double dipping is a very common problem in genomic data science because we often need a complex pipeline for data analysis, and statistical inference is the last step. And our proposal is to use ScDesign3 for generating data from a specified null and use Clipper for controlling FDR without requiring high resolution p-values. And this is so important because it means that we only need to generate the null data for once. So that can save our computational time. So finally, I want to thank my former student Vivian for starting this line of research by developing ScDesign, and Tianyi for ScDesign2, and Dongyuan for the version 3, and also Guangao for the read generating part, and also Xinzhou for the Clipper, and also Kexin for the current integration part, and also the funding agencies. Thank you.
Thanks a lot. We have time for questions. Yes.
Thanks, Jessica. Very interesting. So for the for capturing dependence between genes, have you considered beyond Gaussian cupulas, cupula models that can model asymmetries and so on? And yeah, can you tell us a little bit more about that?
Yeah, so far we haven't. I understand that there can be better cupulas. That the reason is because we found Gaussian cupula worked reasonably well for the data generation, the synthetic, the mimic real data very well. So we didn't actually further optimize that part. But I totally agree that this may not be the optimal choice. But the interesting thing is that since our primary goal is data generation, right? So even though for the, I also want to mention that for the correlation matrix estimation part, one is high dimensional, when no, there will be some issues, right? However, we found that from the perspective of data generation, it can work pretty well for P up to say 2000. Yeah, if we selected 2000 highly expressed genes, it can work pretty well. And we didn't try to those more sparse estimation methods because of computational efficiency. Yeah.
Thank you.
Hi, Jessica.
Hello.
I think I might miss some parts, so I got confused because I think double dipping is a true problem, very serious issue. But I think in your ScDesign3, you still need some cell features or like the pseudo time or the cell type, but they're still calculated. Yeah. So how you avoid?
And the thing is that okay. So I think the key is here. So I think you're right. So in our null data generation, we need to use those features, right? But when we generate the null data, we will try to remove the cell covariates of our interest. So for example, if the real data is classified as or clustered as 2 clusters, and our data generation starts from the 2 clusters. And the reason of that is we want to mimic or estimate the gene marginal distribution and gene-gene correlation, which should be conditional on that covariate. And then when we generate the null data, we will remove the covariate. So that's the advantage of using this probabilistic model, because we can decide which parameters to alter ourselves. And then when we generate the null data, you can see that even though we use the data model fitted from real data, but these synthetic cells are independent of the real data because we are generating synthetic cells randomly by sampling. So therefore, here the comparison is from the first step. So there's double dipping here. There's double dipping here as well. We apply the same pipeline to 2 parts. So if you want to be more rigorous, I want to say that here, when we say the null data generation conditional on real data, we are conditioning, we are conditioning on the real data because we are treating the parameters for generating null data as fixed in this case.
Okay.
Yeah, but as you said, so there's not much we could do for not using the target data cell covariates, because otherwise the null data wouldn't look like real data, right?
I agree.
Yeah.
Thanks.
Hi, very nice talk. Thank you. So my question is actually for the first part, where you mentioned that each gene sort of a univariate distribution could be a Poisson or a zero-inflated negative binomial, anything in between. So does it mean that your copula model sort of, it's a mix of this distribution uniformly?
Exactly.
Okay, and the second question is like, what is the proportion that you observe of genes in a typical data set that is like zero-inflated versus not?
Very, very interesting. Yeah, and we have another paper, which I didn't put here, for showing that result. So if the data are using UMIs, it's UMI-based single-cell data, unique molecular identifier, then I would say 99% we can fit a gene distribution well using Poisson negative binomial. We don't need a negative inflated component. But if it's SmartSeq2, which don't use a UMI, then probably 50% we will use the zero inflated component. Yeah. So in our ScDesign3 paper, although I only show one figure here, but we have many results for different technologies. And this is actually SmartSeq2, I think.
All right, thank you.
Thank you.
Thank you. This is actually, I think, a question almost for both of you. So putting together sort of your patterns on the spatial distribution and putting together your methods for doing the simulations on a spatial grid, I'm curious just sort of your sense of how much of the evidence that's out there is experimental error, how much is really sort of traveling by, say, inheritance of cell type, and how much is real difference in the distributions of either expression or really the sort of isoform question. I'm curious, you must have some way of doing, if not isoform directly, sort of pseudo isoform and the degree to which that's relevant. So I'm just a sort of thought discussion.
Yeah, I think that's a very nice question. And that's aligned with my proposal here for the negative control and positive control, right? So for example, in spatial data, we can fit this model for every slice. And let's say we're doing a comparison between two slices. What we could do for positive control is that we could just generate data without batch effects in our model. So we can know, oh, these genes have no changes for sure. And these genes have changes with no confounding factors from the batches. And we can also have the negative control to include batch effects. We could add batch effects in a gene specific way or universal to all genes. And then we can run the pipeline to see how robust the results are. So these are the things we could do, like in silico experiments with our simulator. Yeah.
So do you have a sense of, how much should you trust today, the results that are coming out on spatial heterogeneity? It's kind of like early on, right? Single cell data is like, yeah, it's noisy and we don't know what to believe. Now we've got some methods that we at scale, you're kind of believing the results. I'm just curious, it's actually incredibly important.
Yeah, I would say it's something we want to investigate further. And I can say that there is, like, I've seen a benchmark paper in bioRxiv for detecting spatially variable genes. Yeah, but I have to.
You don't know, you haven't made up your mind yet.
Right. Yeah.
Thanks, Jingyi, again for your talk. I think that it's time for lunch. We have only an hour today, and we have boxes lunches outside. The other days, the lunch is not provided by Sammons, but there are many places around here. And Ron, I think that we have a picture taking at 2:30 today. Is that correct? In the next break, after the next session, after the break, we'll have a whole group picture taken. Okay, thanks.